Welcome to this year's ALS Therapy Development Institute Summit. My name is John Gang. I'm the Communications Manager here at ALS TDI. And first off, big thank you to our sponsors, Biogen, Cytokinetics, Google, and Mitsubishi Tanabe Pharma. Every year we host the summit to provide an annual community update on ALS research and science, both by ALS TDI and by our partners. And today we are going to cover research in 2021. For those of you who don't know us or might be joining us for the first time, we are the world's most comprehensive drug discovery lab focused solely on ALS. ALS is the only disease that we research. We don't work in anything else. We are also one of the world's only nonprofit biotech companies. We were founded in 1999 by the family of Stephen Haywood. If you look down to the left there at that picture, Stephen Haywood is the man in the orange shirt. He was diagnosed with ALS at the age of 29, and his family were a bunch of MIT-trained engineers, and when they realized there were no treatments out there that could help their son and brother, they decided they were going to do something about it. And so they founded us in their basement in Newton, Massachusetts. And today, our lab in Watertown, Massachusetts, where we are coming to you from, has over 30 scientists working to end ALS. And we put 87 cents of every dollar that you donate to us into finding effective treatments and cures for ALS. And if you look down there to the right, unfortunately, Stephen Haywood passed away in 2006. But in that picture, the guy in the blue shirt is Stephen's son. So this is a family affair, and we are still working to end this disease more than 20 years later. And ALS TDI is unique in the biotech space in that we are the drug discovery engine for ALS. And what that means is that we work to invent and discover drugs and partner to advance them to clinical trials until there are drugs for every person living with ALS. We are the experts in preclinical research for ALS. No one knows more about this disease than we do. We are accountable only to the ALS community. We don't have shareholders. The only investors that we have are our donors who support our work. When a drug succeeds or if it fails in trial, we don't stop. We keep working to find more, and we will keep doing that until there are treatments for everyone living with ALS. And our impact so far, we perform the research required to discover and invent as many ALS treatments as possible. We started the first ALS precision medicine program, which is a partnership with people living with ALS designed to speed up drug discovery, in which we get data from people all over the world living with ALS and use it to inform our research. And if you're watching this right now and you are living with ALS and you are not signed up for this program, we strongly encourage you to do so. We've also rigorously tested over 400 drugs in animal models of ALS, which is more than any other ALS research lab in the world. And we are the first nonprofit biotech company to invent an ALS treatment, AT-1501, and bring it from our lab through FDA review and into clinical trials, where it is now in a phase 2 trial. And first, let's look at the agenda for today. Having a brief welcome right now. At 1:10, we are going to hear from Dr. Fernando Vieira, our CEO and Chief Scientific Officer. We'll then hear 3 presentations on different areas of research at ALS TDI from members of our science team from 1:45 to 2:45. At 3:15, we'll have a 15-minute break. Then at 3:30, we're going to hear an update on Google's Euphonia project, which is a collaboration between ALS TDI and Google to develop communications tools using artificial intelligence. And then at 4:15, we are going to honor members of our community who have gone above and beyond in supporting ALS research for our ALS TDI Research Awards. And then just a little bit of housekeeping. We are going to answer a few questions after each presentation. Won't have enough time to get to all of your questions likely, but we are going to make sure they all get answered. Someone will follow up with you after the event if we do not get to your question. You can submit your questions by putting them into the chat. And now to begin our presentations for the day, I'm happy to introduce ALS TDI's CEO and Chief Scientific Officer, Dr. Fernando Vieira.
Thank you, John. Let me have a moment to set the mic up. All right. Well, thank you all for joining today. Thanks for taking time out of your Friday to learn more about what we're doing at ALS TDI. If you attended our town hall a couple weeks back, there's going to be a little bit of overlap between what I said then and what I'm going to say today. Not complete, but some. And that's because it's a message that's worth repeating. It's about what we're doing and how we're trying to address the problem of ALS. But you're also going to see a lot more throughout the day. You're going to see presentations from some of our scientists who are going to give you in-depth look at the programs that we're running that are trying to get treatments into people. So we all know why we're here. We're here because ALS is a devastating condition. It's a devastating condition that affects the upper and lower motor neurons. It makes those neurons, the motor neurons that are responsible for all voluntary movement, they cease to function properly and eventually degenerate. ALS can cause various signs and symptoms, ultimately atrophy, weakness, ultimately paralysis. There are other signs and symptoms associated with it, but I'd rather focus a little bit more on the nature of ALS, that its effect on the person, right? The person with the disease, the families that are affected, and the friends that are affected, the entire network. ALS ends up robbing people of the future that they planned for. It robs them of the abilities one by one, and it affects everybody in a deep way. So there are some universals to how ALS affects people, but on the other hand, ALS is really quite variable in its presentation, right? So especially at the start, some people might have a variant of ALS that affects the ability to speak first. Some people may have it affect their ability to walk first. Some people may have it affect their fine motor function and their ability to use their hands. And we really don't have a full understanding of that variability. Why does ALS present differently from one person to another? If we delve more deeply into it, we realize that ALS is actually, though there are some universals, it's an incredibly complex disorder that has many different tipping points that might get to that motor neuron dysfunction and motor neuron degeneration. If we subdivide ALS into sporadic ALS, which are the majority, approximately 85% of all cases of ALS are sporadic with no known genetic component, though we believe genetics are involved. And familial ALS, where the disease is directly inherited from one generation to the next, what we find is that there are many, many genes that have been implicated. And this figure, this table that I have here is actually a little bit outdated. Some would argue that there are actually 50 genes that might be implicated in ALS right now. When you delve into what each of those genes do, and we all have many, many different genes, right? We all have about 35,000 genes, and each of them encode proteins that do a job. And when we look at the jobs that the proteins do that are mutated in ALS, we find a lot of differences in them. So what this tells us is there are lots of different initial starting points that can tip, different dominoes that can tip, that ultimately get you to that cascade of motor neuron degeneration. So this is some of the complexity that we deal with. When you go deeper and actually step up from the genome level to the complexity of how cells interact, we see even more variability. Basically, all known human physiology has been implicated in some way, shape, or form in ALS. And That can be daunting, right? That can be overwhelming to a person with the disease who's trying to learn about it. It can be daunting and overwhelming to researchers trying to address the need. But there's also opportunity in that presents us with lots of potential targets, lots of potential ways to go at the disease. When I talk about a target, I'm talking about an intervention point, right? And so while it's daunting, there's also opportunity there. So to summarize these points, yes, we know ALS is a devastating condition that affects a person and leaves them paralyzed and ultimately takes their lives. We also know that it's unexplained in its variable presentation. We don't have a full understanding of that. And that's because of complex biology that's engaged. But that presents us with many possible targets. And so where that leaves us is recognizing the need for multiple treatments. It's going to take more than one treatment to ultimately address the needs of everybody with ALS. And when we look at the current clinical pipeline, there is tremendous opportunity in that pipeline. It's rich. It's as rich as it's ever been. I've been studying ALS for about 20 years. And if you look at the pipeline, there's a lot there. There are a lot of companies involved in the ALS space that weren't there 20 years ago when I started. And there are a lot more drugs available, but it won't be enough. We know that some treatments might prove to be quite effective for small subpopulations of people with the disease, particularly efforts targeting familial ALS, where we know the genes. You may be able to go after them in a targeted way and have bigger effects. Even there, we're seeing that there are challenges, but there's more potential there for those big effects with a single hit. On the other hand, there are lots of treatments in the clinical pipeline today that might have benefits, but they're really on the margins, right? And so you want those marginal wins, you want to take them where you can get them, and you want to stack them on top of each other if you can to have bigger effects. But it does mean, again, it's going to take more than one treatment to do this. It's going to take a lot of treatments. And so the solution is invention and discovery of more treatments until everybody has the options to slow, stop, or even reverse their ALS. And ALS TDI, we're uniquely poised to do that. So we consider ourselves a drug discovery engine that does this over and over and over again to try to find treatments. We do discover and invent drugs in our own lab. We also work with others to validate their own treatments. We partner. When we do invent a drug, we find the partners that can help us get it into clinical trial to make that impact, get it into people. And like I said, and John alluded to earlier, we'll do this over and over and over again until everybody has their needs met. So if you watched the town hall, you saw this figure, but I want to go through it here again today to talk about our role in drug discovery. And in order to do that, I kind of have to explain the drug discovery and drug development process. And so it can break down, there are a few different ways to break it down in a few different words and terms to describe each of these steps, but the ones that I'm using today are to describe fundamental research, drug discovery, lead optimization, IND enabling, so investigational new drug enabling, and clinical trials. And I'm going to walk through those a little bit. So fundamental research, this step is the step that often results in the discovery of that therapeutic target, that intervention point that I mentioned earlier. This is often done in academic research labs that make these fundamental discoveries about how something works in a cell or how something works between cells or at a broader physiology level in a whole animal or in a whole human. The drug discovery step is often the step where you take a piece of this fundamental research and you turn it into an experimental system in a lab that allows you to assess many, many, many different drugs, right? So you're going to hear a little bit later on from our team members today about these different steps, in particular, Kyle, Alan, and Anna. About the drug discovery step, you have systems that will reveal drugs that could intervene and hit the target appropriately and get you to a desired outcome. And you often do this maybe at the 1,000 scale, or even in some cases, companies like Pfizer can do it at the millions scale. Lead optimization is where you take the hits, you take the things that might have been effective during your drug screens, and you put them into a more complex system, a whole animal organism. The whole animal tells you more than cells in a dish can. It really does. Cells in a dish are important, but they don't have a circulatory system. They don't have a liver. They don't have kidneys. They're just giving you a snapshot. So when you put a drug into the whole system, it teaches you more. And in the case of ALS, we're trying to put our drugs into animal systems that have a disease that looks like ALS. And we're trying to make those animals live a better and longer life while not causing harm to those animals in other ways. When you've done that, you have a drug candidate. When you have a drug candidate, right, you then have to make that drug candidate from like this discovery, this invention, you have to turn that into a product that can get into people, that the FDA or other regulatory agencies will allow you to test in humans, in clinical trials. So you have to go through a whole battery of different study types called IND-enabling studies in order to get that done. And once you've done that, once you have that product, then you can move into clinical trials. Clinical trials are tests in people, often with the condition where you're trying to make that impact, to test how it actually is going to behave ultimately in that disease indication. And organizations that run clinical trials are usually a combination of a company that's kind of bill, for lack of a better term, and all of the clinics. And in this case, we're talking about ALS, so we're talking about ALS clinics like MGH, Healy, and Duke, et cetera. So where does ALS fit, or ALS TDI fit in this? So the work that we do in our lab, that's down the hall right here, is the fundamental research, the drug discovery, and the lead optimization steps. That all happens from the 30-odd scientists that are working at ALS TDI every day. Where we partner is at this stage, the IND-enabling steps in the clinical trials. So this is where we would work. We're never going to have a manufacturing facility that's going to make pills, right? That's not who we are. We're never going to have, larger animals. We have zebrafish and we have mice, but we're never going to have, pigs or non-human primates. And ultimately, clinical trials, we would partner with those sponsors or with those hospitals to advance our drugs. So I want to give you a little bit of context for the presentations that you'll be seeing today from some of our scientists. First, you'll be hearing from Dr. Kyle Denton, our Director of Cell Biology, and he's going to be talking to you about some of the processes that go into inventing a drug screen, right? So modifying induced pluripotent stem cells, often from people with ALS, to create that system in which you can test those many thousands of drugs so that you can reveal those hits. Then you will hear from Dr. Theo Hatzapetros, our Director of Preclinical Pharmacology, who will talk about how he takes that handoff from Dr. Denton and ends up he and that team evaluate those drugs in animal models to find drugs that are going to make those animals live a better and longer life, and again, not doing harm. You'll also hear from Anna Gill and Alan Primaseri, associate scientists at ALS TDI, who are going to tell you about our program, our PRMT inhibitor program, where we ended up doing some basic research to identify a new target that hadn't really been looked at in the ALS space before, and how we're moving forward with that fundamental discovery to screen for drugs and then move them forward into those animal models as well. And these are the folks who are going to be presenting today. I wish I could show you every single person on the team. It's an incredible team, and they're busting their butts constantly to make progress. But they're the ones you get to see, and I hope you enjoy it. So ultimately, this whole process, this discovery engine, my objective is to try to advance at least one drug into human clinical development every 2 years. That's actually really ambitious. I know we're talking about the ALS clock, so we do have to push the dial on this to make this happen. But we believe we can. We believe that our pipeline currently could sustain it, and it's our job to continue to fill that preclinical pipeline to keep making this happen until everybody's got their needs met. I also want to give a quick pivot. John mentioned earlier in the introductory remarks our precision medicine program. And I want to talk about that and where that fits in the context of this pipeline. Like, what does that even mean? We have so many people who have been enrolled in this program. It does, like John said, inform our research. We're learning about ALS. But how does that fit into the context of drug discovery and drug development? This is where it sits, where these arrows are marking. So obviously, we don't do clinical trials at ALS TDI, but we can discover biomarkers and measures of disease progression that could be used in clinical trials to maybe make them more efficient. We all know that there are some challenges in the ability to measure if a drug is having a meaningful effect in clinical trials. If we do our jobs right, we could invent and validate digital outcome measures that might be more sensitive, that could notice a more subtle change that's still meaningful than something like ALS functional rating scale. At the same time, we can look for things like blood-based biomarkers that might be more sensitive measures as well, instead of trying to measure, somebody's ability to use their hand to write. Maybe it's enough to measure if there's a change in blood that correlates with a better outcome, and then you can do that in less time and at lower cost. At the same time, we're actually working directly with people with ALS to collect blood samples and skin samples. And those skin samples can be used to generate those induced pluripotent stem cells that you're going to hear about from Kyle today. We use those to develop assay system screens for drug testing. And then finally, this entire effort allows us to identify new targets, new intervention points, maybe not even identify new ones, but helps us prioritize what those targets should be. And so we use those data as well. And this program is the longest running translational research program for ALS in existence. We started in 2014. More than 700 people have participated up to now. Again, I'd encourage anybody with ALS who isn't enrolled right now to go to als.net and go to the Precision Medicine program and try to sign up. We really do. With more data, we can get answers faster. We've had data collected from some folks for up to 6 years. Those are the fortunate people with ALS who have lived with the disease that long. Not everybody can contribute for that long, but we want to learn from everybody. We can learn from the people who have slower disease and see if we can find ways to make everybody have that slower disease. One of the outgrowths of the precision medicine program that we're really proud of is a collaboration that we ended up forming with Google. It was very focused on actually trying to make sense of those digital outcome measures, using accelerometers and using voice recordings to measure disease progression. We've made progress on that. We're excited. We're excited to share those information and data with the community soon. We have a publication that is now on my desk. Actually, I have to hit submit, resubmit. But we didn't expect the fortuitous sort of outgrowth of it, which was the development. It took on a life of its own with the Google team. And I want to tell you a little bit about that. What the Google team realized was that we could use the same machine learning and artificial intelligence techniques, or similar ones, that we were using to assess digital outcome measures could actually be used and deployed to help develop technologies that make it easier for people with ALS today to communicate. So you're going to get an update from a few of the members of the teams who have done that work from Google today. They'll be presenting remotely. Julie Kachow, Lissy Lilienfeld, and Pan Jiang will tell you about some of the progress they've made actually developing products that are available today that people with ALS can use to help communicate. After you hear from Google, you'll also be able to watch the presentation of the 2021 Leadership Awards. And I wanted to just briefly mention the folks who are the inspirations of each of the awards. They were with us during the early years: Stephen Milne, Fran Delaney, Mary Lou Krasnick, and Stephen Haywood. And thinking back to the impact that they had, everything that we've built has been on their shoulders. We're standing on the shoulders of giants, essentially. And people talk about that in the science space. It's the ALS patient community as well. And so it's wonderful that we get to honor people today who have made such an impact and do continue to lead us from the patient and side, but it gives us the opportunity to remember those who did so from the beginning. And so, to summarize, we know the problem, and we know that we need effective treatments for all people with ALS. We know that we can make progress toward the solution. You're going to hear about that today from our scientists. You're going to hear about the power of partnership, that we made a lot of progress because we partnered with Google, and we're excited to see how that's grown and how they've taken ownership of that problem and made a difference. And then you're going to see our inspirations, ALS community members who lead us. And with that, I want to thank you all again for taking the time to be with us today. I also want to personally thank our sponsors, Biogen, Cytokinetics, Google, and MT Pharma, for helping to support this event and helping to support the work that we do at ALS TDI. And with that, I think I'll take questions. Have time?
Yeah. Thank you, Fernando. First of all, I understand that there have been some problems viewing the screen online. We're going to get that fixed right now. So, Fernando, I'm going to ask you to step away from the laptop and come over here with me. First, does anyone in the room have any questions? Okay, we're good. Could you just talk a little bit more about some of the other research organizations that?
There are a lot of different groups that we collaborate with. But most of our collaborations actually come when we're working to validate therapeutics that other companies or infrastructure that allows us to do that. So they'll come to us. They might not have a complete ALS program, but they might have a drug that they think could be impactful. So they'll come to us. They'll often work with our preclinical pharmacology team with Dr. Hatzapetros to assess those. But there are other collaborations we're working with trying to assess biomarkers. Are formal and announced, and with an academic researcher at a university. Do you think we could do anything with that? And then we'll send reagents. And that's an opportunity to do a quick experiment. So that's constant approaches, right? So in the ALS space, particularly in familial ALS. So I still believe, despite some of the challenges here and there, that antisense oligonucleotides can be impactful for some types of ALS. I'm really excited to see, though it seems a bit like black magic to me at this point, how that stem cell research is being done, right? So that we can get a better understanding of how stem cells might work in the context of ALS. Obviously, there are the programs that we've worked on at ALS TDI. So I'm really excited to see how AT-1501 pans out. We collaborated with a group called Neuroimmune that advanced a monoclonal antibody targeting misfolded SOD1. That's now AP101. That's in clinical trial as well. And we also validated in our which doesn't often happen, see how that trial plays out. So those are a few, but the pipeline is promising, and I'm excited about it.
How we decide what to work on in our lab.
We take into consideration multiple facets. I'd say there are three, right? So one, we try to take into account the potential number of people who might be impacted, right? So if we do spend a lot of effort trying to develop therapeutics that might be generalizable to sporadic patient populations. So that's a big piece of it. Another element that we take into account, though, is how much do we know, right? So there is an advantage to knowing that you have a genetic mutation and trying to attack that. So we do have some programs in the past that have focused on SOD1 and currently heavily focus on C9 or somebody else doing it, right? So if somebody will try to attack it from a different angle.
Great. Thanks so much.
Thank you.
Great, thanks.
Thank you everyone for coming in today and for your attention. Today I'm going to talk about our work using induced pluripotent as a series of redox metabolism modulators. From Fernando, ALS is a very complex genes that have been implicated. The participants of our precision medicine program, we have access to cell lines that span all of the, let me just minimize this just in case. A little better. Yeah, so all of the genes here in red, we have access to cell lines. So we can try to study multiple ALS subtypes. Our strategy for studying ALS is using induced pluripotent stem cells, or iPSCs. iPSCs are embryonic-like cells that can be generated from adult cells taken from a patient. So we can take dermal fibroblasts, reprogram them back into induced pluripotent stem cells, and then because they're pluripotent, they can be differentiated into all cells in the body. We're mostly focused on cell types that are affected in ALS, such as the upper and lower motor neurons, so spinal motor neurons and cortical neurons, but also support cells like microglia, astrocytes, and oligodendrocytes. When we have access to these cells, we can study them in vitro or in a dish and use them for phenotypic assays where we look for differences between the ALS cells and the control cells. So a phenotype in people would be eye color or height, but in cells it could be how they survive, their shape or morphology, and other aspects. So when we identify a difference between our ALS cells and control cells, we can use that for testing compounds. So we can do in vitro compound screens where we test hundreds or thousands of small molecules And if we can identify a compound that's producing a beneficial effect, we can follow up experiments on those targets and do hit validation. So we could verify that that hit is a true hit. And if it looks promising, we can pass it on for in vivo testing to confirm that the compounds will work in a more complex system, such as mice or zebrafish. I mentioned that we choose to use the iPSCs because they can generate multiple cell types, and these are just some of the examples of the cells that we are using in the lab. The main, I would say, workhorse cell type is the spinal motor neurons. These are the cells that are located in your spinal cord, and they send their axons out and innervate muscles. And those, along with upper motor neurons, which are long motor neurons that reside in the motor cortex, selectively degenerate in ALS patients. Because there are non-cell autonomous mechanisms or ways that cells can die through interactions with other cell types, we also want to study the cells that are important for supporting the motor neurons, such as astrocytes, microglia, and oligodendrocytes. We also generate muscle cells because motor neurons innervate your muscles, and we can study the neuromuscular junction to look for dysfunction there in co-culture systems. So I mentioned phenotypes in people could be height or eye color, but in cells, there's lots of different phenotypes, but some of the more common ones that we look at are cell survival. So we'll generate cells from iPSCs shown here, from both control and ALS patients, and then look to see whether or not they survive at different levels when we grow them in a dish. We can also do real-time metabolic profiling to look at the energy production of these cells. Protein localization, since a number of forms of ALS have mislocalized proteins and protein aggregates associated with them. Gene expression is another way to look for phenotype that may be different between your ALS and control cells. We do a lot of work on mitochondrial health, and then also spontaneous and induced electrical activity. So we can measure whether or not neurons in a dish are firing action potentials properly and communicating with other cells. Most of our work starts by doing experiments by hand at a smaller scale. So we'll start with like a 6-well or a 96-well plate where you can test 96 compounds at a time. But once we identify a certain phenotype that's interesting, we try to scale up those experiments using cellular automation. So here is a schematic of our automated cell biology environment, which we can sterilize the inside of this chamber, and then the equipment shown over here is located inside, and this is a view from top down. We have this automated cell incubator, so we can hold hundreds of plates at high density formats, all the way up to 1536 well plates, so we can test over 1,000 compounds on one plate. We can send these plates to a plate washer where they get the media replaced, and we can do differentiations without having to be hands-on. We don't have to touch the cells. We just plate them, and then this device will help replace the media. We can then send it to a liquid handler where we can dose those cells with various drugs at different concentrations. And then most of the data is acquired on this automated microscope that we have. So down here is just an example of neurons that were plated and imaged every hour, so we can watch how the cells grow, right now it's starting over again, so we can see the neurites are growing out of the cells. And so we can track those parameters and see if different drugs are having differential effects on some of those. So switching gears to talk about what some of our what we're actually doing in the lab, not just our kind of capabilities. We today are going to focus on our work on SOD1 and C9orf. The reason why is they're some of the more prevalent forms of familial ALS. So SOD1 was discovered in 1993 by Bob Brown's group. This is the superoxide dismutase gene. And then we also have begun working on C9orf72, which accounts for the majority of familial forms of ALS and also 10% of sporadic ALS cases. So starting with C9orf, all of us have this gene on chromosome 9. And we all have 2 copies of that gene, but it was discovered that expansion of this hexanucleotide sequence of DNA is associated with ALS. So most people have 1 to 30 copies of this repeat, whereas individuals where this gets expanded can develop ALS either through a gain of function mechanism where they're generating dipeptide repeat proteins, or loss of function. So you are no longer expressing the proper amount of C9orf protein. And currently the role of C9 is unclear, but evidence suggests that it's involved in cellular recycling, either directly or indirectly. So within our PMP or precision medicine cohort, this is a graph that shows the genotyping data for the C9orf expansion. So the majority of the individuals whose cells we have access to do not have an expansion, so they have between 1 and 9 repeats on both of these alleles. But we do have 21 cell lines with that have a C9orf expansion, and that's helpful to us because with that large number of expanded cells, we're able to overcome the genetic variability that exists person to person. So we can study, we need access to lots of these expanded cells in order to know if a phenotype is a true one or not. In addition to the genome edited into the patient-derived model, we also have been working to generate C9orf knockout iPSCs. So using a CRISPR strategy, we deleted 41 base pairs in exon 2. And when we look at the expression levels of C9orf, you can see our control cells show C9orf expression, but we have a number of clones. So each of these bars is an individual cell line that we've generated, and they're all genetically identical to each other except for this one deletion shown here. And so we have a number that have both copies of C9orf knocked out, and then some that show 50% expression. So they have 1 copy knocked out and the other one is still producing the protein. And this cell line is going to allow us to study haploinsufficiency or the effects of losing C9orf. And we'll compare that data to what we see with the patient-derived cells that have the C9orf expansion. So right now we have access to both the patient-derived C9orf iPSCs and then also the C9orf knockout cells. And we're actively differentiating those into cell types affected by ALS. We're starting with the motor neurons to begin cellular phenotyping. And we also intend to collaborate with our partners in the discovery biology team to look at the effect of PRMT inhibitors on the iPSC-derived neurons. So look forward to Anna and Alan's talk later today to hear more about that. Switching gears to our work with SOD1, we've generated through genome editing a panel of SOD1 isogenic iPSCs. These cells they started with a parental cell line that was had wild type SOD1, so there was no SOD1 mutation. But we introduced a single nucleotide mutation that generated a A4V mutation, so the 4th amino acid was mutated. And this is a highly pathogenic mutation, and there's a severe clinical outcome in patients who have an A4V mutation. So we introduced either 1 or 2 copies of that mutation, and we also at the same time generated SOD1 knockout cells. So you have 1 copy of SOD1 knocked out or both copies. And so this cellular panel can be a source for experiments. So we're using both the edited cells that we generated through genome editing and also those that we have from patients in the precision medicine program. When we have these cell lines developed, I mentioned one of the first things we do is differentiate them into spinal motor neurons. And so we use a method that follows normal embryonic development for the spinal motor neurons. So you start with your induced pluripotent stem cells and then transition them to generate neuroepithelial cells or neural stem cells. They are then further matured into motor neuron progenitors, and then finally spinal motor neurons, which we can use for drug discovery experiments and phenotyping. So an example of one of those experiments, move that a little bit, is shown here. So we do an 18-day motor neuron differentiation protocol, and then plate those motor neurons in a high density format. And then we can either stress them or leave them alone, but then assess their survival daily. So one of the readouts for looking at cell survival would just be imaging the cells, so we can stain them with different antibodies to look at different proteins. So here we're staining for neurites, and then also islet-1, which is a marker of motor neurons. And we can count the number of islet-1 positive neurons that are present in this culture. When we look at the cell survival, down in this bottom panel, these are the wild type on this side, and then the A4V SOD1 mutant motor neurons. We don't see any difference in the number of motor neurons between the mutants and control cells. And in this bottom panel, they have full amount of growth factor present. But if we deprive them of growth factors or almost starve the cells, we see that the A4V mutants preferentially degenerate compared to the control cells. And it also seems like losing one copy of SOD1 is not detrimental to their survival. So phenotypes like this can be used for testing out whether or not we can identify drugs that can prevent this selective degeneration of the mutant cells. In order to test cells with compounds, you need to have access to a compound library. So we partnered with ChemDiv and purchased their target identification library. We chose this library because it has over 35,000 small molecules, all of which have a known activity or target. So if we see that a certain well that was treated with a certain drug has a increased survival in the previous example's case, we can look in a database to figure out what target that compound is acting through, and then use that information to design follow-up experiments to validate that that's an actual true target. So here's an example of some of our compound screening data. Here, we called it compound activity, but this is cell survival. So we have our positive control compounds on the left that are able to rescue the survival of this particular cell line compared to the cells that are untreated. So these cells selectively die off if we don't treat them with any compounds. And you can see the majority of the compounds in the library fail to rescue or elicit any response in these cells. And that's actually a good thing, because if we're seeing most of the compounds rescue survival, that tells us something may be wrong with the design of your assay. And also you wouldn't have the time to follow up all of those hits. Here we can see that there are a few hits that are boosting cell survival, some of them at a level comparable to our positive control compounds. And what was interesting is that we've found 3 of these here all target the same pathway. So that gives us confidence that we should invest more time and energy into following up whether or not that pathway is actually involved in rescuing those cells. And so in order to do that validation, you can either purchase previously published compounds that are known to target that specific pathway, or perform knockdown or knockout experiments to see if that also elicits a similar response as an inhibitor of that pathway would. And so screens like this can give us potential sources of new leads for development. And I mentioned that these 2 positive control compounds over here were able to rescue these cells. And these are actually some of the founding members of a redox metabolism modulators that we've been working with and identified in the lab. So I'm going to talk a little bit about that data in a second, but first, just some background on redox metabolism and ALS. So redox reactions, or reduction in oxidation reactions, have been implicated in ALS, both familial and sporadic forms, for a while. And it's too large of a field to go in depth for such a short talk, but cellular redox states regulate various aspects of cellular function and homeostasis. And so an example of redox, it would be cellular respiration or energy production. And this can lead to the production of reactive oxygen species and nitrogen species. And these can lead to cell damage if they accumulate at levels higher than they should be. And also, elevated reactive oxygen species and nitrogen species have been seen in ALS. And so some of the consequences of redox dysfunction in ALS include mitochondrial dysfunction, ER stress, protein misfolding, and autophagy defects. So this is a piece of early data for us where we were studying an ALS cell line and then also a control cell line. And you can see These are cells in green. This is a panel that has each of these little squares are hundreds of cells, but in green are live cells. And so these ALS cells, when we don't treat or we treat with a vehicle, selectively degenerate. What we found, though, was that when we treat with this one compound that we were calling 268 at the time, it, at a certain concentration, is able to rescue survival of this ALS cell line. We re-optimized the assay and continued to work with it, and we're able to get the cells all the way to the motor neuron stage and then treat with both that original compound and then analogs of that compound and show that we could improve cell survival compared to the negative untreated cells. And this positive group is actually a genetic rescue. So those cells are similar to our wild type cells. We are still actively looking into the mechanism of these redox modulators, but we have seen that they're able to improve mitochondrial health. So if we look at the mitochondrial membrane potential shown here, that's a measurement, one measurement of mitochondrial health. It's generally higher mitochondrial membrane potential is good for cells, and when it becomes reduced, cells can sometimes degenerate, and that's actually seen in certain types of ALS. If we look at our cells treated with these redox modulators, we're able to elevate the mitochondrial membrane potential compared to the untreated cells. And then also we see increased mitochondrial content or biogenesis. So here we have our control and mutant cells at the iPSC stage or non-differentiated state. There's no difference in their mitochondrial content. But when we begin a differentiation protocol on the cells, there's a rapid loss of mitochondrial numbers. But when we treat with this redox modulator, shown here, just two different concentrations, we're able to elevate the mitochondrial content. So this data was exciting to us, and it led to the agreement with ChemDiv for them to generate a series of analogs of the initial compound. And we then re-optimized the assays that we had done by hand previously through our automated system so that we could test them in a high throughput format. These are some of the results from those compound screens where we're testing hundreds of analogs of those redox modulators. And we could see that different analogs are showing different effects on our cell survival here. So some compounds are are more efficacious, so they can produce a higher response in the cells, but others are more potent, so they can work at a lower effective concentration. So by comparing the data from all of the different compounds, we can try to predict which ones will potentially perform better in vivo. And so that's shown here where we're just looking at the EC50 or the potency of these drugs. So drugs with a lower EC50 or effective concentration 50 can rescue the cells at a lower concentration. So the EC50 of the drugs here are below 10 nanomolar. And so we selected the ones that perform best in our induced pluripotent motor neuron system, and then gave those to our colleagues and the pharmacology team. And Theo's going to talk about his work to test some of these analogs in the leading mouse model of ALS. So just to summarize some of the work that we've been doing, we've generated mutant C9orf72 and SOD1 isogenic iPSCs that we are currently using for cellular phenotyping and looking for screenable endpoints that we can use for compound evaluation. We're continuing to run through the target identification library to look for new leads, and then have also characterized a series of novel redox metabolism modulators, which Theo and his group are actively investigating. I'd like to thank all of the past and present members of our team who have contributed to this work. All of these types of experiments are extremely large and require lots of help, so it's definitely a team effort. Definitely need to thank the PMP participants for providing their cell lines, their clinical data, their blood, and their time, because we wouldn't have had access to these cells in order to do these experiments without them. And then I'd also like to thank Augie's Quest for sponsoring the Augie's Quest Translational Research Center, where we're able to do all of the work that you've seen here. And so, yeah, with that, I would like to take any questions you have, and thank you for your attention.
All right, thank you, Kyle. So you focused on C9 and SOD1. How does your work help people with other forms of ALS?
Yeah, so we choose to focus on C9 and SOD1 mainly because the most number of patients have mutations in those forms. But because redox dysfunction is present in many types of ALS, not just C9 and SOD1 based forms, we're hopeful that some of the compounds that we're identifying will be beneficial for both sporadic and familial forms.
Great. Are there any questions in the room? Okay, I think we're good.
Thank you.
Thank you, Cal.
All right, I'd like to call up our next presenter, Dr. Theo Hatzapetros.
Thank you, John. My name is Theo Hatzapetros. I'm the Director of Pharmacology here at ALS TDI. I'd like to welcome you to this year's summit in our new place. And also I'd like to welcome all those of you online. I will pick up from where Kyle left us, and I will talk to you about the in vivo validation and the lead optimization of the redox metabolism modulators that he identified in his assays. And we strongly believe that this can be very applicable for the treatment of ALS. I'd like to start by telling you what in vivo validation is. It is that line of investigation that comes once an experimental therapeutic appears to be promising in cells, but before it goes into humans. It usually takes place in mice, in disease models relevant to the disease you're studying, in our case in ALS mouse models. And we look to see whether a compound is safe, it has drug-like product properties, and it's efficacious in the specific model. And if it does, then it's clear to be developed for IND to develop enabling studies. And if it doesn't check any of those boxes, it's either trashed, or if it has desirable properties, it might be repurposed for another indication. We have done a lot of in vivo validation studies at ALS TDI. As a matter of fact, it's the only thing that we did since day one of the Institute. Over the last 20 years, we tested around 750 experimental therapies. And these include drugs or small molecules, as we say, protein biologics, gene therapies, stem cell therapies. Not all of them were tested in survival efficacy studies. They were not deemed suitable for further development, but we have conducted over 500 survival efficacy studies in the mouse model. These are lengthy studies. They take anywhere from 4 to 6 months. They take a lot of animals, and they're designed to determine if a drug is efficacious in an ALS model. Most of the therapies, unfortunately, fail in this step, in the in vivo validation step. In fact, only 2% of the experimental therapies were validated in vivo. 2% is a low number, but it's good to have the number below. It means that we set a very high bar for success, and we use a very stringent method of validating a compound. But there is an explanation why it is so low, and there's two reasons for it. The first reason is that our ALS mouse model is a hard model to rescue or to make better, as it should be, since ALS is a very complex and hard disease to treat. Many of you know that we use the BLAC6 SJL SOD1 G93A high copy mouse for these studies. we like to use this mouse model, this particular mouse model, because its clinical presentation is very similar to that of ALS, that of both familial and sporadic ALS. They show there is motor neuron loss in these mice. There is muscle wasting leading to paralysis. It is a progressive disease. There's astroglial and microglial activation. The inflammatory gene expression profile of these mice is very similar to that of humans with ALS, and there's decreased survival. It's also conducive to drug screening, what we do in this mouse, in that, unlike other models, the disease onset is relatively early. It happens after about 90 days. The disease progression is moderate, giving us the chance to intervene therapeutically and perhaps make these mice live longer. There is some variability, but it is acceptable, meaning that the mice between each other, they behave relatively similarly. And finally, it's been around for a while, so people recognize this model as an applicable model for testing ALS therapies. In our hands, this model has been quite successful in predicting the outcome of human studies. Almost all of the therapeutics validated in this model fall into one of three categories. They're either drugs that have been approved already for the treatment of ALS, like riluzole, therapies that are currently in clinical trials for ALS, like AT51, that we discovered it in this institute, now in clinical development by Eledon Therapeutics, copper ATSM, also in clinical development by Medicinal Collaborative Medicine, right? And AP101, initially developed by Neuroimmune, now licensed to ALS Pharma. The third category is there are compounds that are currently quite promising, and they look as if they will advance soon in clinical development. And one of these group or class of compounds are the redox metabolism modulators that Kyle talked about earlier. And I will also expand on those later. The second reason why our validation rate was low, frankly, has to do with the way we've been selecting compounds in the Institute. So in the early days of the Institute, when Jamie Haywood was trying to find a therapy for his dying brother, Stephen, they would simply test all those molecules that seem promising through published reports, many of them poorly designed published reports. And they would test them in the mouse, and they would simply look whether it increased their survival or extend or delay their onset. And most of those attempts were unsuccessful, unfortunately. Nowadays, we select compounds in a much broader way. If you can see on this graph, we have different sources of selecting compounds. Some come through collaborations. Collaborations is something we strongly believe in. Our mission is to find therapies for ALS. Sometimes we might need to help others find therapies for ALS, and that's very consistent with our mission. We also repurpose drugs that coming from other disease indications with similar mechanisms as ALS, so MS or Alzheimer's. Finally, we have medicinal chemistry capabilities. Now we can synthesize analogs of promising drugs. And finally, is what Kyle mentioned, through multi-compound libraries. There's one library that he's screening right now that contains 35,000 compounds. So we have a bigger pool to find clinical candidates from. And we also have more readouts to test whether they do what they're supposed to do. We still rely strongly on onset and survival, but We use metrics like CMAP. CMAP is an EMG technique used to assess the neuromuscular function of mice. We use histological endpoints to see whether drugs improve mice at a cellular level. We use gene expression studies, and finally, assays like ELISA to look at protein levels, or Simoa platform to look at neurofilament levels. And finally, the SOD1 mouse is no longer our only model to model this disease. You've heard from Kyle that we have human iPSC cells, and you'll hear from Anna and Alan that we have other mammalian cell lines to test drugs, and these processes talk to each other and they inform each other, and sometimes it might go from the mouse back to the human iPSC cell line and back and forth. So by conducting our research this way, we have better informed compounds, and as a result, we have more hits. A hit, I define a hit as a compound therapeutic that produces significant benefit in a mouse model by either making it live longer by extending the survival or by delaying the onset of the disease. This graph shows you the frequency of hits over the years. I'm sure you can appreciate that in the early years of the Institute, we didn't get many hits. As a matter of fact, it took us 5 years until we found a compound that worked, that was riluzole, and then we would get a hit every other year. Sometimes we wouldn't get a hit for 2 years. Well, more recently, there's a sudden explosion of hits. So over the last 5 years, we've been getting a lot more hits, which is a good thing. Most of these recent hits is from an emerging class of compounds that Kyle referred to as redox metabolism modulators. Several of them have already produced significant preclinical benefit. The exact mechanism that these drugs work is still being actively investigated, though we think that redox modulation is implicated. Redox stands for reduction and oxidation reactions, which are physiological processes, but the dysregulation of which has been described by some in the etiology of ALS. From these molecules, TDI 1692 is the most advanced so far. TDI 1692 significantly delayed disease onset in the mice by 16 days and extended their survival by 16 and a half days. This might not seem a lot to you, but keep in mind that these mice only lived to be 120 days. So it's 15% life extension. And also keep in mind that their disease only starts at 90 days. So it's close to 30-40% slowing down of the disease. So 1692 significantly slowed down disease progression. It also improved neuromuscular function, as assessed by CMAP. And very importantly, the positive effects of 1692 were observed repeatedly in both genders and at different doses. It's fair to say that 1692 is our best performing drug ever in the SOD1 G93A model. 1692 has outperformed other successful drugs. Cooperative TSM and anti-CD40 ligand were the two most successful drugs we've had before testing TDI 1692. These are Kaplan-Meier curves showing survival. Cooperative TSM increased the median survival in mice by 5 days, anti-CD40 ligand by 9 days, while TDI 1692 almost tripled the extension seen with covalent TSM and almost doubled the extension with anti-CD40 ligand, producing a 16.5-day survival extension. 1692 also improved the neuromuscular function as assessed by a technique called CMAP. We've only started using CMAP over the last 2 years. That's why I don't have a comparison with the other compounds. CMAP indirectly measures the amount of healthy axons in a mouse. In ALS, in human ALS and in mouse ALS, as the disease progresses, these axons die off or they malfunction. And as a result of it, CMAP values go down. So the first group on the left are mice that did not receive any drug, and all of them showed a decline in CMAP, about 18 units, while all of the drugs that received TDI 1692 on average did better. So some mice did not decline at all from baseline, some actually became better. TDI, we consider TDI 1692 to be a possible but not a perfect lead candidate. It has 2 caveats associated with it. One, it has a narrow therapeutic index, which means that the concentration or the dose producing efficacious results is too close for comfort to the dose that causes adverse effects or toxicity. It's not unheard of for compounds with narrow therapeutic index to make it into clinic. Warfarin is one of them, levothyroxine is another one, but it's not a desirable characteristic per se. The second caveat with 1692 is that its solubility is less than optimal. It's not as bad as a rock in solution that I put there, but it takes some effort to make it go into a solution to formulate it for animals. This formulation would not be acceptable for humans. Thus, our lead optimization efforts have led to the development of a series of compounds structurally related to this one. These 14 redox metabolism modulators have already been scaled up for in vivo testing. All of them are performing very well in Kyle's assays, and the work is to determine their pharmacological compound properties is currently ongoing. And our goal would be to identify a compound with equal or better efficacy than 1692, but better pharmacological and pharmaceutical properties. TDI 1831 is our newest favorite from this series. It shows improved solubility compared to 1692. It is better tolerated than 1692, and it produced significant extensions in onset and survival of about 7 days. We hope that with the necessary dose optimization studies, we will increase its efficacy or produce extensions similar to 1692, which was around, or better, which was around 16 days. I want to leave you with 2 messages. The first one is that the pool of preclinical candidates that we currently have under in vivo evaluation is bigger and better than ever before. And secondly, to tell you that our objective is to advance a new therapy into human clinical testing for ALS every 2 years. Fernando said this in his opening speech. And we believe that we might have the first one of these compounds in the lead molecule from the RMM series. I'd like to acknowledge the work of many people that they did for this program. Anybody from people handling the animals, caring for the animals, designing experiments, doing the analysis of experiments, IT support. Everyone has contributed largely to this project. And I will take any questions. Thank you for being here and for listening to me.
Thank you, Thea. First question from the Zoom chat. Why not use a TDP43 mouse model? Wouldn't that be more broadly applicable to people living with ALS?
We'd love to use a TDP43 model. We'd love to use as many models as possible. We think that our model is quite applicable to people with ALS. It might be a transgenic model, but in terms of downstream mechanism and clinical presentation, it looks very similar to that of human ALS. So we think that there is translatability there. But I agree that the more the models you have, the better the outcome will be. And TDP43 is a model that interests us. C9orf models are also interesting. The problem is to find a model that has those phenotypes that are relevant to ALS.
And are other models like that being developed?
Yes, in addition to testing drugs, one of our focus is to develop alternative models of ALS. It's not always easy to find one. We've tested tons of TDP43 models. Unfortunately, none of them look like human ALS. They present some features of ALS, but not all of them. We continue this process currently. We bring new models all the time. Whenever we cannot find a mouse model, we bring zebrafish or other organisms. I think we plan to bring Drosophila soon.
And maybe just as an example, can you talk a little bit about what riluzole looks like in a mouse survival and efficacy study? Like how many days that life is extended and stuff like that?
Yeah, I didn't show riluzole on purpose because compared to covalent TSM, CD40 ligand, and 1692, the extension was very modest. It was only 3 days. So any of the 3 compounds that I showed perform much better than riluzole in our animal model.
And are there any questions in the room?
With the mouse models that you guys use, do you use any of the mice? Are they cloned versions, or are they all individual, unique mice?
So the transgenic mice, so genetically, they're very similar to each other. They're bred together. What's different between them is that the amount of SOD they express may differ from mouse to mouse, and as a result, the disease progression is slightly different. We try to control for that. We genotype them, and we only allow mice to enter our experiments that fall within a range of gene copies that Val can tell you more about. She's sitting right behind you. But we try to make them as similar genetically as possible. In addition, we divide them into, we split every litter of mice into a control group and treated group. We thus controlling for the gene variation further, meaning the brothers and sisters are separated in half of them are drug treated and half of them are not.
And any other questions?
I'm just curious, or I just want to be clear on the 1692. As you led us through that, it was sounding so promising, and then you got to the point where like that led us to a different group with related characteristics or whatever, and I understand that whole piece. But is 1692 then gone, or is there study that still is going on with that compound?
Good question. 1692 is not gone, and in the event that we don't find a compound that is better than it, it can very well be a lead compound. It's just a little bit harder because of those two undesirable characteristics that I told you to move it into clinical development. But there are other similar compounds that are approved for human use right now. So I wouldn't say that it's gone. It's very much alive.
Anybody else? Okay. I think we're good there.
Thank you. Thank you.
All right, thank you very much, Theo. And next, I'd like to welcome up Anna Gill and Annalyn Promisseri.
I'm much shorter than Theo. Okay. All right, so hi, everybody. Welcome again to the ALS TDI Summit. My name is Anna Gill, and I'm an Associate Scientist 2 here at ALS TDI. And with me is Alan Promisseri, our Manager of Clinical Operations, and a fellow Associate Scientist 2 as well. So today you've heard from Dr. Vieira, Dr. Denton, and Dr. Hatzipetros about several of our research initiatives here at ALS TDI. And now Alan and I are going to walk you through another project where we're developing type 1 PRMT inhibitors as a novel therapeutic target for ALS. So I'll start with an overview of a bit of the basic biology behind this project, including what PRMTs are, and then I'll move into how they might be connected to ALS. After that, I'll dive into why we started studying PRMTs, including some of the data that indicated to us as researchers that their inhibitors might be a promising therapy for this disease. And finally, Alan will walk you through how we are currently moving this project forward to bring us closer to using them as a potential treatment. Sorry, it's not advancing. Did I click it? Oh, okay, sorry. So this project is primarily focused on protein biology, specifically how proteins act and how we as scientists can manipulate that behavior. As you can see in this diagram, proteins are made up of subunits called amino acids. These amino acids are linked by peptide bonds to form a polypeptide. This polypeptide then undergoes post-translational processing, including things like splicing and folding, to form a mature protein. Mature proteins make up roughly 20% of the total composition of the human body, and subsequently they contribute to crucial processes in the muscular, nervous, and immune systems where ALS pathology takes place. Once you have a mature protein, it can then be altered in several different ways, some that are reversible and others that are irreversible. Each of these changes, known as post-translational modifications, are crucial to determining things like how a protein functions and where it localizes in the cell. So as you can see in this diagram, post-translational modifications of a target protein can include the addition of structures like polypeptides, chemical groups, and complex molecules, as well as changes in amino acids and protein shortening or cleavage. In our project, we focus on the addition of chemical groups, specifically methylation, as a way of manipulating a protein's function and location like an on or an off switch. And so what are PRMTs and why are we looking into them specifically? PRMTs, or protein arginine methyltransferases, are proteins that carry out post-translational modifications on other proteins. Specifically, they modify the amino acid arginine by methylating the nitrogen atoms that make up an arginine's net positive charge. All PRMTs can monomethylate an arginine residue, meaning add one methyl group to one of the two nitrogen atoms. Specifically, type 1 PRMTs, what we are looking at, can add a second methyl group to that same nitrogen atom. We call this asymmetric dimethylation because the methyl groups are unevenly distributed across that net positive charge, kind of asymmetrically to one side. Conversely, Type 2 PRMTs can add a methyl group to the second nitrogen atom on the other side of the positive charge. We call this symmetric dimethylation because the methyl groups are then evenly distributed symmetrically across that net positive charge. So why do we care? What does this have to do with ALS? PRMTs carry out the post-translational modifications that I just described to you on a large body of proteins. And as you can see in this diagram, these are proteins involved in vital cellular processes, such as RNA processing, splicing, and translation. Now, if I highlight all the proteins in this web that have been implicated in ALS research already, you can see that PRMTs in fact modify, meaning change the function or location of a substantial fraction of the known ALS-related proteins. And this gets us interested in seeing how manipulating this activity could change ALS protein pathology. And so why did Alan and I specifically start looking into this? As Dr. Denton mentioned earlier, C9orf72 mediated ALS is facilitated by a hexanucleotide repeat expansion mutation in chromosome 9, open reading frame 72. And this is the form of ALS that Alan and I were studying when this project started. Patients with C9 ALS exhibit 30 to thousands of repeats of this G4C2 sequence, whereas healthy individuals have between 1 and 30 repeats. These extra repeats in C9 patients cause the resulting RNA to be very unusual in shape, having what we call hairpin and quadruplex structures, as well as areas that translational machinery may mistake for a place where it should start or stop making a protein when that sequence isn't actually there. These make it very difficult for that machinery to make the native C9orf72 protein that all of us have. And it makes it much more prone to creating mutant species of protein in its place. And so as a result, there are 5 known mutant proteins that have been identified, resulting from translating those abnormal RNA structures. And they are known by their amino acid composition, like I mentioned earlier. They are called poly-GR, PR, PA, GP, and GA. These proteins, derived from that highly repetitive RNA sequence, manifest as repeating dipeptides. And so they are known as dipeptide repeat proteins, or as Alan and I prefer to call them, DERPs. Okay. So we have studied what happens to cell viability when we apply the DERPs to neuron-like cells called NSC34s. This graph shows the amount of LDH released from NSC34 cells. LDH is a common protein found in almost every cell in the body. So when cells are sick, the integrity of their cell membranes decreases, and LDH can leak into the surrounding media. We can then detect this and quantify how sick cells are after we challenge them with various conditions. Thus, in this graph, the higher the point that you see, the higher the percent of cell death in that sample. And so what we consistently observe is that the DERPs containing arginine, poly-GR and PR, are consistently more toxic in a significant manner than the DERPs that do not contain arginine. And this brought us back to how we know that PRMTs can modify arginine residues. Could the PRMTs also modify these mutant proteins that are toxic to cells and somehow change that toxicity in a way that can help us find and develop new treatments? And so we went to the lab and started to try to figure this out. To determine if PRMTs could modify these mutant GR and PR proteins, we performed a series of reactions in tubes where we combined the type 1 PRMT, PRMT1, a source of methyl groups, and either its known substrate, histone H4, or poly-GR. We then performed Western blot analysis to look at what occurred during those reactions by using antibodies to detect either that histone H4, poly-GR, or asymmetrical dimethylation, so that we could detect if the PRMT was adding a methyl group to those proteins. And so our controls behaved predictably. When histone H4 was present, it was detected. Same with GR. But interestingly, we found that GR did appear to be asymmetrically dimethylated with an increasing dose of PRMT1 applied. Furthermore, this was even picked up by the histone H4 antibody, which specifically looks for an asymmetrically dimethylated arginine residue on histone H4. So basically another GR that is naturally occurring. In summary, GR was being methylated by a PRMT, and 2 different antibodies were seeing this dose-dependently. And so once we confirmed that PRMT1 could alter the toxicity of, or could alter the C9 ALS protein poly-GR, we then wanted to look at how this affected the toxicity of the protein. When we applied either asymmetrically dimethylated GR or symmetrically dimethylated GR to neuron-like NSE34 cells, we found that asymmetrically dimethylated GR was significantly more toxic than its counterparts at doses from 100 nanomolar to 30 micromolar. The margin, as you can see on this graph, is of course quite narrow, but we believe that this could result from some of the unmethylated GR we applied being modified by type 1 PRMTs after it enters the cell, based on what we saw in the blot data that I just showed you all. And so this led us to ask whether in C9 ALS, these type 1 PRMTs could be modifying GR and PR to make them more toxic, and we wanted to see if we could somehow mitigate this using small molecules for potential treatments. And so we tested this theory, and it had very promising results. When we administered the type 1 PRMT inhibitor TDI 1804 to those NSC34 cells, after we challenged them with either GR or PR, after 24 hours, the 1 and 3 micromolar doses of TDI 1804 completely rescued dysmetabolism induced by either of the peptides. In these graphs, the decrease in metabolic activity induced by GR or PR challenge is indicated by the decrease in absorbance between untreated and vehicle controls and the white GR or PR bars, as you can see here. You can then see the 2 preceding bars to the left, 2 doses of the type 1 PRMT inhibitor TDI 1804 restore the height of those bars to equal the healthy controls. This is a massive deal in the lab. You often will not get complete rescue with a given compound, and this is the result that was really encouraging to us to move this forward. So we saw these results repeat at those 2 doses for several different cell viability endpoints. And we were encouraged to then see if they would extend to other small molecule PRMT inhibitors. And so we proceeded to test various commercially available small molecule PRMT inhibitors in our GR and PR challenge system using neuron-like NSC34 cells. We found that across all cell viability endpoints, 5 inhibitors rescued toxicity to varying degrees. This is represented in this diagram by how large the circles are. Inhibitors in a larger circle rescued the most effectively. This kind of allows us to narrow which structures are the most effective as well. Interestingly, all of the inhibitors that showed rescue effect were type 1 PRMT inhibitors, and all of the type 2 PRMT inhibitors that we tested had no effect, similarly to our negative control compound TDI 1813. The only type 1 PRMT inhibitor that had no effect also had no significant activity at the endpoint that we were testing. So its lack of effect was still consistent with our previous results. The amount of times that this rescue phenomenon occurred using different small molecule inhibitors gave us the push that we needed to really dive into this project and learn more about the biology behind the rescue effect we were observing. And so, as a result, we've published extensively on this body of research over the past 3 years. In 2019, we published a paper in the International Journal of Molecular Sciences detailing the cell viability assay system we developed that I run to assess toxicity and different rescue effects. And then during the height of the pandemic in 2020, we then published our therapeutic indication in Frontiers in Pharmacology. This 2020 paper got a lot of attention in the research community, so this year we wrote a follow-up published in Frontiers in Cellular Neuroscience, where we dove deeper into the biology we think is at play and commented on how we might use this research to develop more promising treatments. And so now that you guys know the history of the project, its impact, and all of our contributions, I'm going to hand it off to Alan, who will describe some of the steps we're taking currently in the lab to advance this. Thank you.
Cool. Thank you, Anna. And I will take it over from here and give you a broad overview of where we're going with this project. So this is ongoing work now. As Anna detailed, we have found a biological target and have found some beneficial effects of using these drugs in cells that are in the presence of the dipeptide repeat proteins, or DERPs. In the process of finding a potential treatment, all that work fits under fundamental research and drug discovery. In fundamental research, we find a target of interest, try to understand the mechanism of action, and develop an assay or an experimental setup that allows us to test compounds targeting that mechanism. The screening of potential treatments in that assay system is the drug discovery portion. The next step would be lead optimization. Some of the work that Theo outlined earlier would fit in that category. And it includes determining how these compounds behave in a more complex model, like an animal model. Of course, it's hard to know exactly how a molecule will behave in an animal when you're starting out in a cell. During lead optimization, we can find limitations of the drug we're working with. And what we found with the PRMT inhibitors from earlier is that they had trouble entering the central nervous system in mice. Getting distribution in the CNS means the compounds need to pass through the blood-brain barrier, which is this layer of cells that's pretty selective in the molecules that allow to pass through it into the central nervous system. So what we need now is a PRMT inhibitor that is similar or has better activity than the ones we were testing, and also be able to pass into past the blood-brain barrier. This does mean we need to take a step back and design a compound that can achieve both goals. Now, to find a new compound, we enlisted the help of a lead discovery company that is capable of designing compounds and testing them in an in silico screen, which is a virtual or computer-based screen. And what they'll do is they'll take the crystal structure, which is what this diagram is showing, of a PRMT that is complexed with an inhibitor that is known to work, and they'll use that to guide their design of thousands of compounds. Then using different models and prediction software, they can get a sense of which molecules will perform the best. The advantage of a computer-based screen is that they're able to design and test thousands of compounds in little time, but the obvious disadvantage is we're not dealing with the actual protein and the actual compound. However, we can collaborate with a company like this to test a variety of chemical structures that may improve on the compounds that we are currently working with. After receiving the compounds from our partners, we have to test these potential treatments in our own hands to see if they work as advertised. This will require each compound to pass through different model systems before we can move it into lead optimization. And so the first that I'll quickly outline for you is a simple test of asymmetrical dimethylation inhibition. And in this assay, we just start with a 96 well plate, and with each well is coated with a PRMT substrate. In this case, it's histone 4 that Anna mentioned earlier. And when we add the PRMT to that well, they will methylate that substrate, and after a few other steps, it'll give off a luminescent signal. So it's pretty quick and easy. Now, we take the test compounds, and when we add them, ideally, they should inhibit that methylation from happening, and therefore there should be no signal that's coming out of that. And we can test multiple compounds at a time with this assay in one experiment. And because our collaborators are providing us with a lot of compounds, we need a system like this to quickly filter out potential hits and move them on to the next stage. And so when we find strong performers in that previous assay, we move them into the viability models that we've established. And here I'm just quickly showing you the battery of cell health measures that Anna alluded to earlier that we look at and that have all been negatively affected by the addition of the DERPs and then rescued by the addition of the PRMT inhibitor. And we use some of these in the work Anna described earlier, and they measure various aspects of cell health, including how they process different molecules, how they live, proliferate, grow, and die. And once they move through that, once we find molecules that can move through that as well, then we'll be able to move them into lead optimization and tweak them how we need to until they enter the blood-brain barrier and get into the central nervous system, as I described earlier. We'll also be we'll be doing this in mice, and again, as Theo mentioned earlier, we have other models that we would like to test these in. We can test them in the iPSCs that Kyle had mentioned, and we do have a C9 zebrafish model that we have in-house, and we're looking to expand into a fruit fly model that is also C9 related to help just bolster the data and really support our findings. Now, I do also want to mention that all this work doesn't mean we stop the fundamental research necessarily. While we're focusing on the drug discovery and getting to that lead molecule as fast as we can, we are continuing to explore how the inhibitors affect PRMT biology. As you saw earlier with this interactome, or as some of us like to call a radiculogram, because it's just a crazy web of all the proteins that could be affected, there's a lot of pathways that PRMTs are involved in. And now it would take a lot of work to just start exploring other pathways. So it does give us another chance to look externally and see what we can learn from the rest of the community. So recently, this group published a method where they evaluated in one big experiment the transcript, the expression of transcripts of like thousands of transcripts and levels of like 700 proteins in cells that were exposed to a PRMT inhibitor. And so they were able to get a sense of what was affected when those PRMT inhibitors were added. And so we're hoping to adapt this method into our work, have the cells be treated with the DERPs or with the PRMT inhibitor and see how that affects the cells in different ways. And we're hoping to find new targets out of this sort of assay and not necessarily related to C9. PRMT research before this has demonstrated that they're interacting with other proteins like FUS, TDP43, and other RNA binding proteins that are not necessarily related to C9orf72. And I won't delve into it here, but our team is doing a variety of other experiments to help us further our knowledge of what those mechanisms might be. This is helpful for making future research decisions, and it's also helpful when developing a lead drug to know the extent of the pathways that are affected, as it helps us understand what subtypes of ALS the drug might be best suited for. So to summarize, we found a potential therapeutic target in PRMT inhibition, and we're working off of our findings from last year to design and evaluate an optimal compound or compounds that could be our lead molecule or molecules, while still expanding our knowledge about PRMT mechanisms to uncover new targets in C9 and non-C9 related ALS. I hope at the very least, this has given you a sense of the process of how a compound comes from the very early stages of research and moves through a variety of tests and assays and obstacles for sure to move through that pipeline that Fernando outlined earlier and to eventually hopefully become a lead drug. I'd like to acknowledge the other members of our, we call us the PRM team. And they're, as I mentioned, they're doing a lot of other work that's just as important, although not outlined here, to help us get to that goal of new targets, understand what's happening. Val and Kaylee specifically doing bench work. Fernando just tells us what money not to spend and when to spend it. So yeah. All right. And with that, thank you all for listening to our presentation. We'll take questions.
Thanks a lot, Alan and Anna. So first off, are there any questions in the room? Okay. So can you talk a little bit more about the PRMT inhibitors themselves? Maybe tell us a little bit about their chemical structures.
We did call them TDI, whatever, in this presentation, but they're actually all available. Their structures, they're commercially available already. So if you look up our papers from last year, you'll be able to find out what they are and what their structures are and what they've been used in. They're not drugs already, but they are compounds that have been explored.
Got it. And you use what you call neuron-like cells. What is meant by that, and why did you choose those type of cells versus, say, the type of cells that Kyle is using?
So the NSC34 cells are a very versatile cell line. Basically, they start as a hybrid line that mimics the spinal cord, but they still grow at a pretty rapid pace. So we can do a lot of assays without spending a long time. But if we want to look more closely at the biology once we find kind of a hit, we can then differentiate them using different chemicals to make them more like a mature motor neuron that would be in a patient, and then we can study it further. So it kind of acts as a way for us to not waste too much time on drugs that aren't hits, but still be able to look closely at the biology when there is a hit.
Awesome. Thanks so much, guys. Unless there's any other questions? Okay. Thanks a lot. All right. Big thank you to everyone from the science team who just presented their work from the past year. Now we are going to take a little bit of a break. We are running a little bit ahead of schedule, but we'll still plan to come back at 3:30. All right, 3:30? Yep. Cool. All right. So we will see you in a little bit.